Hey gang, welcome to Microbiology for Surgical Technologists. I'm your host, Tiffany Bailey, and I've been teaching with the college for 18 years. My field of expertise is clinical laboratory science and microbiology. And like I said, I've been doing this for a long time. So hopefully you enjoyed the course. We're going to be learning quite a bit in the next 16 weeks or so. So stay tuned for more information on Unit 1. There's several broad objectives that I want to try to cover in the next 16 weeks. First of all, I want you to have an understanding of what it means to have a sense of surgical conscience, and I want you to avoid causing harm to your patient. We always got to avoid causing harm to our patient. So in other words, we have to make sure that we're not introducing microbes to our patient in the surgical suite. I want you to outline types of microorganisms and their ability to cause diseases, list mechanisms for the spread of pathogenic microorganisms, correlate the impact of microbiology on infection control in the operating room, compare the structure and characteristics of different microorganisms, and finally, I want you to analyze immune responses that occur in the body as defense mechanisms against pathogens. So there we're going to talk a little bit about immunology. Some of the topics in microbiology that we're going to cover will be microbial growth and nutrition. We're going to talk a little bit about bacterial biochemistry and metabolism, bacterial genetics, host-parasite interactions and disease transmission, significant isolates versus commensal flora, the study of viruses, which is virology, the study of funguses, which is mycology, and then finally we're going to cover disease processes. So that's kind of what you can expect in the next few weeks. There are several pioneers in microbiology that I want to talk about today, including Girolamo Fracastoria, which was an Italian doctor from the 1500s, and he suggested that a disease comes from clotting of particles, and he defined syphilis as a disease. The next person I want to talk about is Robert Hooke. Robert Hooke lived as an English scientist in the 17th century and studied cork under a microscope, and he called each segment of the cork a cell. Then I want to talk to you about Anton van Leeuwenhoek. Anton van Leeuwenhoek is a Dutch scientist. He was actually a lens maker, and he called bacteria under the microscope the wee beasties, or the animalcules, and he was alive in the 1600s, the late 1600s. Okay? He first noted bacteria under the microscope in 1673. Louis Pasteur is no stranger to science. Pasteur developed the germ theory of fermentation. Bacteria turn wine into vinegar. Low heat killing of bacteria in milk is called pasteurization. Then we have Robert Koch. Robert Koch was a bacteriologist that came up with what's known as Koch's postulates. So in other words, if we have a disease in one animal, it can be transmitted to another animal, and then the bacteria can be cultured, and the same bacteria will grow in the new culture. And then we have Joseph Lister. Joseph Lister was a pioneer in surgery in the 1860s. He came up with the principle of aseptic technique. He believed strongly in wearing gloves and gowns and sterilizing instruments. And there's a picture of Joseph Lister right there on the right. Let's talk about some hot topics today in microbiology. Coronavirus was first its claim to fame really came in the 2020s. Since that time, 1.9 million deaths have occurred due to a respiratory disease, and there is a vaccine now available for coronavirus. Usually that includes a couple initial vaccines and then some boosters. The Middle Eastern Respiratory Syndrome, that has caused 145 deaths worldwide, but only 2 in the United States, and both patients actually were from Saudi Arabia. The Middle Eastern Respiratory Syndrome is another respiratory disease. Measles is on the rise, and that's due to the lack of vaccine users. There was actually 13,000 cases in the United States in 2019. The Ebola virus was big news in 2014 when there was an outbreak that killed 11,000 people. The University of Nebraska Medical Center has a certified biocontainment unit, and they actually treated 3 patients for Ebola in 2014, and of those 3 patients, 1 did die. We should be concerned with resistant organisms. Gram-positive cocci are now very resistant. I'm sure you've heard of MRSA, which is methicillin-resistant Staph aureus. Some of our gut flora, like Klebsiella pneumoniae, is an extended spectrum beta-lactamase producer, so it's very resistant as well. And then another thing that is a hot topic in micro today is the cholera pandemic. We are currently in a pandemic with 5 million cases a year occurring every year. You have to have a good understanding of the chemistry of life in order to understand microbiology. Now, Earth is actually 4.5 billion years old, and we have to talk about the carbon cycle, the nitrogen cycle, and the sulfur cycle. Now, in the carbon cycle, carbon is limited, and carbon is constantly being recycled by carbonobacteria, which is going to oxidize carbon monoxide to carbon dioxide. All living things are made of carbon. In the nitrogen cycle is needed to make nucleic acids like DNA and RNA. The nitrogen fixation traps atmospheric nitrogen and in turn can become plants, and it can be used by plants and animals. And the sulfur cycle is needed for the formation of amino acids. Amino acids are made into proteins, and bacteria release sulfur from hydrogen sulfide, making sulfates. The carbon cycle, the nitrogen cycle, and the sulfur cycles are needed to make amino acids, lipids, carbohydrates, and proteins. There are two domains I want to talk about. One is called archaea, and one is called bacteria. The extremophiles live in the archaea group. They are single-celled, rely on lipids and cell membranes. They often are extremophiles, like I mentioned, and they are commensal organisms of the human microbiome, and DNA is not contained inside of a cell nucleus. The thermophiles are extremophiles that love really hot temperatures, and the halophiles are ones that really love salt. Okay. We also have the domain of bacteria, which are simple, single, small organisms. They are prokaryotes, and they have very few complex organelles. They do have nucleoid, ribosomes, plasma and cell membranes, pili and flagella that helps them adhere to surfaces and gives them options for motility. And they are grouped together by genetics and metabolic activity. They either live singly or in communities known as biofilms. Prokaryotes versus eukaryotes. Let's go ahead and talk about that. So the prokaryotes are small and simple. They have few complex organelles, and they live singly or in biofilms. They have membranes, cytoplasm, DNA, ribosomes, pili, and flagella. Bacteria are prokaryotes. We group bacteria together based on morphology, staining, motility, metabolism, and genetics. The eukaryotes are usually larger and are usually larger than about 10 microns, okay, all the way up to humans, so they can get very large in size. They have more complex organelles. They have mitochondria, which makes fats and carbohydrates. They have endoplasmic reticulum, Golgi bodies, which stores proteins, and ribosomes, which makes proteins. In this slide, I want to go over the structure of prokaryotes. Okay, first of all, they have a cell wall that is rigid and gives the cell its shape. Gram-positive organisms, which we're going to talk about later, have a thick peptidoglycan layer, and gram-negative organisms have a thin peptidoglycan layer, and they also have pores in their cell wall. They have cytoplasmic membranes. Here they secrete toxins. They have cytoplasm, also known as endoplasm or ectoplasm. That's going to be the inner and outer cytoplasm with cytosol. Cytosol is the fluid that has the nutrients and the waste in it. Okay. We have capsules. Prokaryotes sometimes have capsules, and those capsules mean that the organism cannot be phagocytized by our white cells. And those capsules are made from a substance known as glycocalyx. We have chromosomes and plasmids. Plasmids, of course, are small circular chunks of DNA. Prokaryotes can have flagella, which is a means of locomotion. They can have pili, which helps an organism stick to a membrane, or they can have what's known as the conjugation pilus, which moves DNA to the daughter cells. Some prokaryotes have spores, and other prokaryotes have ribosomes, and ribosomes make proteins. Now, there's 4 different arrangements of flagella on certain prokaryotes. We have one flagella on one end of the cell body, and that's known as monotrichous. We have one flagella on both sides of the cell body, and that's called amphitrichous. Then we have tufts of flagella on both sides of the cell body, and that's called lophotrichus. And finally, we have flagella all around the cell body, and that's called paratrichus. Nutrients and waste are transported across cytoplasmic membranes, and that can happen by either passive or active transport. In passive transport, we have a substance that moves from an area of high concentration to the area of low concentration without the expenditure of energy. This happens in three ways: diffusion, where substances move from a high concentration to an area of low concentration. We have osmosis, where molecules of living cells in water move from a high concentration to a low concentration. And in osmosis, we have isotonic solutions, hypertonic solutions, and hypotonic solutions. In isotonic solutions, water moves in and out of the same rate. In hypertonic solutions, the solutes are outside and are then moved inside the cell. And in hypotonic solutions, the solutes are inside of the cell and move to an area outside of the cell. Then we also have osmosis, okay? We also have filtration. Filtration is when you have hydrostatic pressure from the heart beating that pushes molecules across the membrane, okay? We also have active transport. Just keep in mind that in passive transport, there's no use of energy here. They're not using ATP at all. In active transport, this is where a substance moves from an area of low concentration to an area of high concentration, and it uses an expenditure of energy. So here we are using ATP. There are 4 terms associated with active transport, including endocytosis, exocytosis, pinocytosis, and phagocytosis. In endocytosis, we have solutes that are grabbed with an expedition of energy and brought inside the cell. In exocytosis, the content of the vacuole is released outside of the cell. In pinocytosis, this is what I refer to as cellular drinking, and the fluid is brought inside the vesicle. And in phagocytosis, this is cellular eating, where solids are brought inside via a vesicle. So here in this slide, I basically break down passive transport a little bit more. We've got diffusion, filtration, and osmosis. In diffusion, substances move naturally from an area of high concentration to an area of low concentration. In filtration, we have hydrostatic pressure that pushes molecules across a membrane, and usually that has to do with the beat of the heart. We have osmosis, where molecules move across a membrane where there's a high concentration of living cells in water. And then we have those 3 solutions: isotonic solutions, hypertonic solutions, and hypotonic solutions. So that's a little bit about passive transport. In active transport, we have a substance that moves from an area of low concentration to the area of high concentration with an expenditure of energy. And we can break that down into a couple of terms. One term is called endocytosis, where the cell uses energy to bring something inside the cell. Pinocytosis is cellular drinking, where it's bringing fluids into the vesicle. Phagocytosis is cellular eating, where solids are being brought into a vesicle. In exocytosis, the contents of a cell are released to the exterior of the cell through fusion. So bacteria need 3 things to be viable and reproduce. They need carbon, nitrogen, and a source of energy. Okay? A carbon source would be carbon dioxide or a sugar like glucose. Minor needs of the bacteria are sodium, potassium, calcium, and chloride. Bacteria also need nitrogen, and bacteria also need a source of energy, which is usually ATP. So you're going to find out in this course that every organism has a very long name that's usually hard to spell. Naming the bugs is called nomenclature, classification, and taxonomy. Okay? Nomenclature is to name an organism. Binomial nomenclature means you have a 2-term naming system, generally refers to the genus and species. So for E. coli, that is a binomial nomenclature. We would say it's Escherichia coli, and that you can see there that refers to the genus and species name. Classification is dividing the organisms into groups, And taxonomy is the combination of classification and nomenclature based on genetic makeup and an organism's phenotype. An organism's phenotype is an outward expression of something. Let's go ahead and look at the taxonomic classification of Escherichia coli. The kingdom would be bacteria. The phylum would be Proteobacteria. The class is Gamma Proteobacteria. The order is Enterobacteriales. The family is Enterobacteriaceae. Anytime the word ends in ACA, it refers to the family. The genus is Escherichia, and the species is coli. Notice how we capitalize the genus name, and then we write out the species name all in lowercase, but we always want to make sure that the entire genus and species name is either underlined or italicized. An excellent way to preliminarily identify bacteria or fungi or even parasites is by using microscopy. We have several different types of microscopes in the clinical laboratory. We have a compound light microscope, that's also known as a bright field microscope. We have a dark field microscope that can be used to identify things like Treponema pallidum, which is syphilis, where we look for the light-colored spirochetes on a dark background. We have phase contrast microscopy, which bends the light through the ring-shaped condenser and puts a halo around things that have a low refractive index. We have fluorescent microscopy, which uses a fluorochrome like fluorescein isothiocyanate. And then we have electron microscopy, and there's two different types of electron microscopes. There's a transmission electron microscope and a scanning electron microscope. In the transmission electron microscope, electrons are held to a very linear, narrow beam of light through the specimen. In a scanning electron microscope, electrons hit the entire surface of the specimen, and we can actually magnify something up to 10,000 times. And then here you can see all the different parts of the microscope. Starting at the top, we have the ocular lenses. Those are also known as the eyepieces. Sometimes you have just one eyepiece, and that's called monocular, and then other times you have two eyepieces, and that's called binocular. Each one of these oculars will magnify a specimen 10 times. Okay? Below the ocular, we have the revolving nose piece that allows the head of the microscope to be revolved backwards and forwards. Underneath the revolving nose piece, we have the objective lenses. That's the most important part of the microscope. Those objective lenses are usually objectives that magnify the specimen either 4 times, 10 times, 40 times, 50 times, or 100 times. And if you take those objective powers and you use them in combination with the oculars, you would actually be able to magnify something 40 times, 100 times, 400 times, 500 times, or 1,000 times. Below the objectives, we have the slide holder, which is also known as the mechanical stage. Below that, we have the stage. Below that, we have the condenser, and on the condenser, we have a little lever known as the iris diaphragm. And then below the condenser, we have the light source. On the side of the base of the microscope, we have the light intensity control, which is also known as the rheostat. Above that, we have 2 knobs. One is called the coarse adjustment knob, and one is called the fine adjustment knob. And then at the very back of the microscope, we have the carrying handle, which is also known as the arm. Using a microscope does require quite a bit of practice in order for you to really perfect that skill. Okay? One of the first things we're going to do is we're going to take our slide and we're going to put our specimen on the slide, and we're either going to heat fix it or we're going to fix it in methanol. And we do that so that our sample doesn't slide off the slide. Then we place the slide on the stage, and we take that 10x objective and we get it as close to the slide as possible without actually touching the slide. You can actually adjust the intrapupillary distance and the diopter. The intrapupillary distance allows you to see one object instead of seeing 2. It just basically brings both your left eye and your right eye into focus, in addition to the diopter. The diopter allows you to focus your left and right eye. Okay? The intrapupillary distance has to do with how far apart each ocular is based on your pupillary space. Okay? Make sure your condenser is centered. Okay, and then what you want to do is take your course adjustment knob and back away from the slide as a little bit at a time as you're looking through the oculars. Once the object comes in focus with the coarse adjustment knob, then it's time to use the fine adjustment knob to sharpen that image. Turn the light up and then go to your 40x objective. Use your fine focus and turn the light up a little bit to get the object into focus a little bit better. When it's time to go to the 100x objective, you can add a little bit of oil to your slide and then snap in that 100x objective on oil. Again, turn your light up a little bit and use your fine focus, and objects should be parfocal. And what I mean by that is any object that you see on 10x, when you go to 100x, it should be relatively in focus. Here's just a nice YouTube video on how to use a microscope. Now, most stains used in the clinical microbiology laboratory are differential stains, but occasionally you will use a simple stain. A simple stain looks at the morphology of an object only. It doesn't really tell us much about their cell walls or any of their organelles. There is no contrast provided using a simple stain, and it may just tell us if an element is either present or absent. For example, if you use the methylene blue stain, you could tell if you have a white cell or a red cell based on the methylene blue stain. There's a red cell. There's a white cell. There's a number of different differential stains that we use in the microbiology laboratory. The one that we use primarily and most frequently is called the Gram stain, and that's named after a man by the name of Hans Christian Gram. Okay? We also have acid-fast stains. Organisms that will not decolorize very well with decolorizer made with acid are called acid-fast. And there's a couple different types of acid-fast stains we can use, and we'll talk about those later throughout the course. Those are called the auramine and rhodamine stain and the Kinne's carbofusion stain. We've also got endospore stains, which stains the spore, capsule stains, which stains the halo of the capsule around the organism, and then we have flagella stains, which actually stains the flagella, or their anatomical part for locomotion. Let's go ahead and take a look at the Gram stain. The Gram stain looks at the peptidoglycan layer of a cell membrane, and organisms can be either classified as gram-positive or they can be classified as gram-negative. A gram-positive organism is an organism that has a very thick peptidoglycan layer in its cell wall, and you can see that in the image on the left. And then our gram-negative organisms are organisms that have a very thin peptidoglycan layer, and they're also very porous. Okay, we're going to use 4 reagents to stain these organisms. We're going to use crystal violet, iodine, alcohol acetone decolorizer, and safranin. And then when we use a series of these 4 stains, the resulting look of your bacteria are either going to be purple. Those purple organisms are called gram-positive, or they're going to be pink, and those pink organisms are called gram-negative. So let's take a look at some of the reactions on the next slide. So organisms that retain the purple stain, also known as crystal violet, organisms that retain that are purple. Now, organisms that have a thin peptidoglycan layer and are very porous, they will be decolorized with the acetone alcohol decolorizer. And so what happens is the gram-positive stain, the purple stain, actually flushes out of the organism, and they are left only having a pink color with the counterstain of the safranin. All right, so let's take a look at some of our gram-positive, gram-negative organisms. So up there is a squamous epithelial cell. That's nothing we need to worry about. That large, irregularly shaped cell is just a squamous cell. A squamous cell are the types of cells that line the inside of your mouth. And then over to the right, we have a white cell. You can barely see the nucleus of the white cell, but the darker pink areas of that cell is the nucleus of the white cell. And then finally, we have gram-positive bacteria, which is our purple-looking bacteria. And then we have gram-negative bacteria, which is our pink-looking bacteria. Some bacteria don't gram stain very well, and so the best thing we can do is do what's known as an acid-fast stain. They have waxes in their cell walls that allow them to stain with the acid-fast stain. There's two different types. One is a bright field stain called the Kinyan's carbofusion, and then there's a fluorescent stain known as the auramine-rhodamine stain. In the bright field stain, the Kinyan's carbofusion, you can see that organisms are red on a bluish background. This is about a 10-minute procedure. On the right, we have the fluorescent stain. This is called an auramine-rhodamine stain, where organisms are bright orange on a black background. This uses a fluorescent microscope and does require about 30 minutes to do this procedure. Other differential stains are not typically used in a clinical microbiology lab, but it might be something used in research. We have three stains known as the endospore stain, the capsule stain, and the flagella stain. The upper right image is an example of the capsule stain, and you can see a halo around the gram-negative organisms. The middle image on the left is a flagella stain, and you can see the paratrichous flagella coming out all over the cell body. And then finally, on the bottom right, we have a spore stain where the spore is green inside of a vegetative cell. Some organisms, such as Clostridium and Bacillus, are going to stain with malachite green, and then the red stain is acid fusion. Bacteria can be described as three shapes, either cocci, where they're spherical, bacillus or rods, where they're elongated, kind of like a cigar, or spirochetes, where they're spirals. And I've given you some examples of each bacterial shape. Our cocci would include Staphylococcus and Streptococcus. Our rods would be Escherichia coli and Salmonella typhimurium. And then our spirochetes would be Campylobacter, and then Treponema, which causes syphilis, and Leptospira. Let's take a look at what some of these bacterial shapes look like in the next slide. So because all of these organisms are purple, we could say they are gram-positive. The image on the upper left is cocci. They're spherical. This would be a Staphylococcus where they're in clusters. On the upper right, we have gram-positive rods. You can see they are bacillus-shaped and they are staining purple. And then finally, in the bottom image, we have spirochetes. And you can see they kind of got the little wavy cell bodies. An example of that would be the syphilis organism, Treponema pallidum. Let's talk about culture media a little bit. This is a base for which to grow bacteria on it so we can identify the bacteria. They are made primarily of agar, which is a complex polysaccharide media. The medias can be very basic, and that's called basal media, or it can be very complex as well. Let's look at the types of media in the next couple slides. Types of media that we use in the microbiology lab would be things like enriched media, selective media, differential media, anaerobic media, and transport media. A type of enriched media would be 5% sheep's blood agar, and you can see an image of that on the upper right. Okay. Also, other types of enriched media would be PEA and CNA. We'll talk more about those later. An example of a selective media would be MacConkey agar, and it's also differential. And you can see a MacConkey agar on the bottom right. That pink colored media is an example of MacConkey agar. Then we have anaerobic agar, which is something that has biosalts and a pH indicator. It helps show, it's also got hemin and vitamin K in it, which helps us culture anaerobes. Anaerobes are organisms that don't require oxygen. And then we have transport media, which is basically a holding media that promotes the viability without actually promoting multiplication of the organism. So all of that media that I talked about before is good to grow prokaryotes. Now, if you want to grow viruses, we have to use what's known as cell cultures. Okay? Cell cultures are living cells that are used primarily to grow viruses, and there's lots of different types of viruses that we can grow on cell cultures, including herpes virus and respiratory viruses like influenza or RSV. We can use certain types of tubes to grow our viruses, and those are called roll tubes or shell vials. A roll tube is a long test tube that is kind of rounded on the ends, and a shell vial looks like a tiny little vial that holds what looks like a contact lens. Okay? We can use all different types of living media, including monkey kidney cells, rabbit kidney cells, human fibroblast cells, And anytime a virus has affected those cells, we can see what's known as cytopathogenic effect under the microscope. So let's take a look in the next slide what cytopathogenic effect is. So this is rhesus monkey cells underneath the microscope, and you can see that in the left image they are not affected. You can see the cells are not swollen at all. And then if you look in the middle image, they're starting to swell, and then if you look at the right image, that is an example of positive cytopathogenic effect, where the cells have really swollen, and that indicates they are infected with the virus. Now, organisms have to grow in the correct atmosphere or they won't reproduce or be viable. Some organisms are known as obligate aerobes, so they absolutely have to have oxygen. One example of that is Pseudomonas aeruginosa. We also have obligate anaerobes. These are anaerobes or bacteria that absolutely cannot have any oxygen. Then we have our facultative anaerobes, which are organisms that grow anaerobically. That's kind of preferred, but they grow just as well aerobically with oxygen. An example of a facultative anaerobe would be Staphylococcus. Then we have our capnophilic organisms. These are organisms that require 3 to 10% CO2. An example of that would be Haemophilus influenza. And then finally we have the microaerophilic organisms. These organisms prefer 85% nitrogen gas, about 5% oxygen, and about 5 to 10% CO2. One example is the stool pathogen known as Campylobacter jejuni. So there's a number of relationships that microbes can have with its host. One is called mutualism, one is called commensalism, and one is called parasitism. In mutualism, both the organism and the host benefit. In commensalism, one benefits, but the other is not harmed. And then in parasitism, one benefits and one is harmed. So let's talk about the process of infections. We have a couple different types of organisms that live on or within our body. One is called resident flora. These are normal organisms, and usually our normal organisms outnumber the pathogens about 10 to 1. They're not easily washed off with hand washing, and these are kind of the good guys. We want them to live on or within our body. In fact, we have about 3 pounds of microbes living on or within us at any given time. Transient flora live temporarily on us. They're usually not bad guys, but they can become bad guys. They can be opportunists, and they are easily washed off with sheer forces. Okay? An opportunistic pathogen is when the body's immune system fails and the organism cannot colonize and starts to unleash its virulence. I know that sounds scary. Some examples of opportunistic pathogens that you may have heard of before would be Pneumocystis, Cryptosporidium, and Mycobacterium kansasi. And when I think of these organisms, I think about HIV patients because a lot of these organisms can be very bad opportunistic pathogens for people that have AIDS. Pneumocystis causes what's known as pneumocystis pneumonia in HIV patients. Cryptosporidium is a sporozoea that can cause problems for immunocompromised hosts. And Mycobacterium kansasi is an acid-fast organism that can actually colonize people that are immunosuppressed. Commensal flora is normal flora. These are normal organisms that actually compete with pathogens for minerals and nutrients. They do play a beneficial role within the human body. They can actually break down bilirubin to urobilinogen. They can actually make vitamin K. We actually want normal flora on or within our body. Now, normal flora can be found on the skin, the upper respiratory tract, the gastrointestinal tract, and the female genital tract. So let's talk about bacteria in all of these body sites. There's quite a number of different organisms that are normal flora on the skin. We've got Staphylococcus. Sometimes it could be Staphylococcus aureus, but usually it's what we call coag-negative staph. These are staphs different than Staphylococcus aureus. We also have Streptococcus, but not the same type of strep that causes strep throat. We have Micrococcus, which are similar phenotypically to your Staphylococcus. And then we have Corynebacterium dipteroides, or these are called dipteroids. These are gram-positive rods that usually live in the environment. Okay. For example, if you culture a cell phone that's been touching your ear, it's going to grow out normal flora because you have normal flora that lives on your ear. The upper respiratory tract includes the throat, the nasopharyngeal area, and the nares. Okay, this is not the lower respiratory tract. There should be no normal flora in the lower respiratory tract. But in the upper respiratory tract, we can see organisms like Staphylococcus, Neisseria, Streptococcus, Rothia, Haemophilus, which is a tiny gram-negative rod, typically not Haemophilus influenza, however, and then Lactobacillus, which is a gram-positive rod. Okay, and these are all normal organisms of the upper respiratory tract. In your gut, or in the gastrointestinal tract, there are many hundreds of thousands to billions of bacteria. Anaerobes outnumber aerobes about 1,000 to 1. The type of flora you're going to see in the gastrointestinal tract would be miscellaneous anaerobes, lactobacillus, diphtheroids, enterics, staphylococcus, and enterococcus, which is essentially a streptococcus of the gut. Entero means gut. Now keep in mind there's no normal flora in the stomach because the stomach has a pH that is inhabitable for most bacteria. It usually has a very low acidic pH. Now in the large intestine there's hundreds and hundreds to billions and billions of bacteria. Normal flora of the female genital tract would include lactobacillus, staphylococcus, streptococcus, and diphtheroids. Lactobacillus is actually a benefit to the female vagina because it keeps the vagina at a pH of 4.5 because of the production of lactic acid. Normal flora of the female genital tract that keeps the vagina at a pH of 4.5 helps prevent pathogens from growing there because most pathogens cannot tolerate an acidic environment. A biofilm is a community of organisms living together and acting as one. All of these organisms can essentially communicate with one another about things like being resistant. They all live together on a surface as a community. So what are the pathogen reservoirs? Where do pathogens live before infesting or infecting the host? Reservoirs can be many things, including inanimate objects or living things. Some pathogenic reservoirs could be plants, insects, animals, and humans. Okay. Other reservoirs that are non-living would be air, soil, and fluids. Transmission of an infection can go from an infected person to a non-infected person in a variety of ways, like direct contact, like touching something and not using good hand hygiene. That's how you can get a disease. Sneezing or coughing, secretions such as kissing or sexual contact, blood and body fluids. I always think of hepatitis B and HIV. Or contaminated food or water. Here I think about things like hepatitis A, or water sources or food sources that are contaminated with human feces. Now, how an infection or an infectious agent gets on or within the body is called a portal of entry. Skin can be a portal of entry. For example, Staph aureus can go through the skin and cause our patient impetigo. Another portal of entry could be mucous membranes, including the mucous membranes of the respiratory tract, the gastrointestinal tract, and the genitourinary tract. In the respiratory tract, if the mucous membrane is damaged, you can catch things such as influenza virus. In the gastrointestinal tract, if the mucous membranes are compromised, you can get things such as Clostridioides difficile, also known as C. diff. All right, so that's it for Unit 1. We've covered a lot of things, including the chemistry of life, portals of entry, different types of stains, normal flora. So if you have any questions, please reach out to me. You can give me a call at 437-2761, or you can email me. That's probably the best way to get a hold of me. I usually respond pretty quickly at tbailey@southeast.edu. Thank you for your attention. Have a great day.